Well, thank you. Actually, the title was wrong in the program book. I'm not focusing on neotropical palms. And today, as my abstract reflects, I'm going to be talking about phylogeography of palms, and I'm going to address this at 3 different taxonomic levels. And so I just want everybody to relax and take a little bit of patience with me. Hopefully my talk is some thought process and perspective on phylogeography today. Because for those of you who know me, actually, I come from a very different perspective. I started my work in palms on population genetics specifically. I did quite a bit of systematics and phylogenetic theory with my advisor during my PhD, as well as some empirical work on palms and macroevolution. And it's really just now that I'm starting to work on phylogeography of palms during my postdocs. And I started with Carlos Jaramillo at STRI, and now I'm currently working with Alessandro Antonelli. I have 2 large-scale projects focused on phylogeography. So today this talk will really just function as getting all of you up to date with the projects I'm working on and giving some personal perspectives on phylogeography. These are personal perspectives, and I'm certainly no expert in this field. So just to outline then briefly what I'm going to be talking about, I just want to get us all on the same page about what exactly phylogeography is. I don't think that many people here in the audience are actually working on phylogeography outside of our colleague Julissa Roncal. So I think it's important that we all get on the same page. Secondly, I'll be starting at a broad taxonomic level, and I'll be talking about how biome shifts affect species diversification and phylogeographic patterns. I'll then be talking about how geographic distribution is affected by symmetry and phylogeographic patterns, and I'm going to break that into 2 different tales or stories from my work. All of this today is unpublished, so I'm hoping to get everybody inspired to do some new research. I'm going to talk first about some interesting results we have on Southeast Asian congenerics, and then I'll be talking about this Western Amazonian bagunza, or mess, that we see here in the neotropics. And then I'll finish up very quickly talking about next generation sequencing, and future prospects in phylogeography. So then let's all get on the same page, take a sip of our coffee or our water, and just relax and think a little bit exactly about what phylogeography is. So we can define it then as the study of historical processes that are responsible for contemporary geographic distributions of individuals. And this contrasts then with the study of biogeography, which we've seen quite a bit here in this conference, which is a field of study that's more focused at the species level. But very intuitively, there's a lot of crossover between phylogeography and biogeography. And it was John Abies who made major contributions and really generated the science of phylogeography. Most of his work were based on fish populations, and most of them actually were not wild populations that he worked on, but he forged the way for this new field of science. It was his seminal book in 1987 entitled Phylogeography, together with advances in the coalescent, that brought us this field called phylogeography, which is really population and phylogenetic perspectives on biogeography. So there haven't been that many papers specifically on phylogeography in palms. We saw a fantastic new study by Julissa Roncal, but one of my favorite studies so far in phylogeography, specifically on palms, was published last year in the Journal of Biogeography. And it really embodies what we should all be striving for, and represents cutting-edge research at the front of palm research. And so those from Goiânia, Brazil, published this paper focused on Meritia flexuosa, where they coupled phylogeographic and demographic simulations with ecological niche modeling and the fossil record to understand patterns of phylogeography in Meritia flexuosa. So as I said, they married different data sets. They had demographic simulations, pollen information, and then they did a large phylogeographic study to understand how glacial cycling affected population size in the Amazon through time. And what I think is one of the most important parts of this study, in my personal opinion, is the dense sampling they were able to achieve throughout the Brazilian distribution of Meridia flexuosa. They found high differentiation amongst populations, and this was a really wonderful contribution to phylogeography in palms. So while the De Lima et al. Paper was in review and in press, a grant was approved from the Brazilian government to understand these same phylogeographic patterns, but across a continental scale, across South America. And so myself, Rosane Colevaci, and Alexandre Diniz Filho are currently working on understanding patterns across the Amazon basin. We can see here depicted in red are those points that I just presented from the De Lima paper, and all of these green points here represent populations that I've collected over the last nearly 6 years that I've been living in Colombia. And so we also have our sequence capture data back from using an Illumina platform across these populations. And we've recently hired an Atalaya expert who just finished her PhD with Jean-Christophe and Andrew Henderson at the New York Botanical Garden, Cynthia Gomez de Freitas, who's based now in Guyana with Alexandre Diniz Filho and Rosane Colevaci, working on our sequence capture data. So what we're interested then is looking at tropical forests through time. And we don't only want to use our sequence capture data, we want to marry this and harness the power that's involved, or the power that one can get when comparing genetic data with the fossil records, such as from pollen. So what we're interested then is population size through time, particularly in the Quaternary. And so one would imagine that you would have in interglacial periods wetter conditions, and therefore you would have increased forest tracts or higher distributions. And during the dryness of glacial times, you would have then contractions of Meridia populations and, as a proxy, tropical forests. The other interesting thing that we want to do with this project is try to understand patterns in recent times and then model reactions to future climate change projections. So we have made some progress. Most of this is coming from the fossil pollen record through collaborations with various people. So first off, in the Amazon fan region, we've been working with Karina Horn, who's now just produced the review of the Meridia fossil pollen record from the more eastern parts of Amazonia. So then these data will be compared with our data using coalescent methods to look at the differences in abundances through time. And so not only are we interested in one region of South America, we also want to compare then what's happening in the Amazon fan region to that which is happening in the Llanos region of both Colombia and Venezuela. Through collaborations with STRI, with Carlos Jaramillo, as well as colleagues at Ecopetrol, which is a Colombian oil company, we aim to also review the pollen record available for Meridia in the Llanos region. So not only will this give us a better perspective of Meridia flexuosa through time, but it's also going to be a very interesting comparison to look at the Llanos region versus the Amazon fan. So all this is well and good, but I think that really the power that we need to start thinking about with phylogeography is looking at this in a comparative way. We can integrate across various taxa or various regions then to understand more general patterns through time. We saw how powerful this tool was in a recent paper by Juan Carlos Santos, who looked at dendrobatid frogs across the neotropics. This is a fantastic study and very powerful indeed, where he showed various examples of Indian lineages filling niches in Amazonia, showing these general basic comparative patterns that I think that we should all be striving for in the future. It's wonderful to have specific case studies, but I think we might get more statistical power when we compare different types of taxa. So now that I've given more of a more personal perspective of exactly maybe what phylogeography is and where we should be going with phylogeography, giving a few snippets and examples of what's going on with palms and with phylogeography in general, I'd like to talk a little bit about another project that's very much in progress about biome shifts affecting phylogeographic patterns in species diversification. So what exactly are biomes? Biomes then we can define as major ecological communities or complexes of communities. And it's been well recognized that each biome has its own taxa and therefore its own history. There's been quite a few recent publications coming out of our lab at looking at just this. And really though, I think the most important paper still remains one from Mike Christ from 2009. And what he did was look at general patterns across the entire southern hemisphere. And what he found was that speciation events occurred in the same biome or between similar biomes, and that actually shifting biomes was quite rare. And he found of nearly 11,000 speciation events in his data set that only just over 350 of them were shifts. And so I'm mostly interested in this to go back to some of the questions that Thomas Couvreur and Bill Baker spurred me to think about with their Origin of Tropical Forest paper, and to further examine the function of dry ecosystems in palm evolution. And so there are various lines of evidence that we can use to understand biome evolution. We can use climactic models, like some of our colleagues, Pierre Sepulchre in France, is working on, where you use current projections and apply them to the past. But I'm certainly not an expert on modeling climate. So then I can refer to two other different tools that we can use to understand biome evolution. And those include fossils and phylogenies. And they likely give us similar predictions, where the age of biome-specific lineages are going to tell us something about biome evolution, such as those examples I just gave. Another prediction from these two types of data is that we can then think about tropical forests, and we can think that with increased that we get increased dispersal with forest connectivity. So when there's warmer climates, we might get more rainforests and more connectivity amongst them. And when we have cooler climates, we see the evolution of drier areas and reduced connectivity. So then, what we're interested then in doing is developing large phylogenies. Specifically for me, of course, I'm very interested in having a species-level phylogeny of palms. But there's been a lot of work done by Soren Fosby and his colleagues already. But what we're planning on doing is, or what we are currently doing, is using two different tools. We're using our super smart bioinformatic platform, which has been developed in my lab, and we're comparing that then with a phylogeny derived from gem-based sequences. So I'm not going to speak about the super smart platform today, but if you're interested in reading more, you can come and talk to me, or you can refer to our website or our GitHub site to see more. So just really briefly here, we have made advances. Here's the GenBank tree. It currently includes 808 species, strictly from MAC-K and RBD2 sequences. And this phylogeny includes a lot of unpublished data, for example, from colleagues such as Angela Cano, and unpublished sequences of my own as well. And this contrasts with the very large tree that we're able to get from SuperSmart, and we're also able to include many more markers. And so then to code these biomes, what we're doing is, on the global level, we're using the World Wildlife Foundation biome classification. These are some things that are going on in my lab, but I'm mostly focused on the neotropics. So I had great ambition to show you some really interesting results, particularly when I found out that Soren already had a species-level phylogeny published, but things come up. Maybe you've seen Mia going around with my mother-in-law, and also my co-advised PhD student, Alexander Ziska, also just had his baby girl in late May. So instead, what I've decided to do is then just show you some general patterns that we've been deriving in my group that we will then apply specifically to palms. So this is work that is coming out of our lab in various different publications. So we're using these phylogenies to analyze biogeographic histories across taxa at a phylogeographic scale. So when we look at these different organisms, what we can see is when we break them into biomes, we can use stochastic mapping to look at connectivity amongst them. So I think this is going to be a really exciting tool to look at palms both in the neotropics and at a global scale. So to actually show you something besides just ideas, I thought I would show you some of the results we have in review on angiosperms as a whole. We're using Amy Zane's large tree of over 5,000 species, and we have good coverage across the 7 different biomes that we're coding in South America. And then we need to go back to the different data sets that we can use to understand biome evolution. And again, I said that phylogenies and fossils have similar predictions. And then we can refer to this interesting paper that Carlos and Andres Cardenas published in late 2013, where they showed some very interesting patterns on South American rain versus wet biomes. What they showed was that tropical rainforests actually did not expand to temperate latitudes during warming events, and that instead non-analog systems evolved or emerged, and that this tropical rainforest diversity there increased, and that it appeared to be a function, or could be a function of temperature, which also Fabienne Condamine has also shown. So to complement those fossils, we're finding very similar patterns then using phylogenies. We found that when we break into our different dry biomes, for example, and where we have our dry north and our dry diagonal, we also see no significant increases in dispersals during global cooling events, as you can see here. And when we then refer to wet biomes, when we have the Mata Atlántica here and the Cuenca Amazonica here, we see that when we look at dispersal events through time, to examine the connectivity between those 2 biomes, we see these few results. And this would be correlated to with the evolution of this Cerrado biome. What we're finding is we see no significant increases in dispersal through these times. And we see no significant drop, as I just mentioned, reflecting the origin of this dry biome between the 2 regions. And we are suggesting that this is then supporting the fossil record, where we think novel biomes or non-analog systems are forming. So overall, our results for this example in angiosperms are showing that Caribbean angiosperms in general are mainly derived from the Amazon, not from Central America. And I think this is a very novel result. And that the Atlantic forest is serving as mainly a source or a potential refuge, which Ana Carnaval has also shown in frogs. We are showing that the Andes exchanged more with Central America than the Amazon, which is likely due to also the position of the Choco. And then we're showing that the Amazon is both a source and a sink for angiosperm diversity. So, I think that these types of tools are going to be very powerful for looking at different taxonomic scales in palms. And so I want to then move on, keep this talk going, and talk a little bit about symmetry and how symmetry might affect phylogeographic patterns, particularly in palms. I'm going to start off talking about this lovely tale of Southeast Asian congenerics. I'm so delighted that John Dransfield can be with us here today to see these results in person. So this paper then is called The Evolution and Maintenance of Symmetry in Tropical Forests. And of course, as an evolutionary biologist, it always tickles you to just be able to throw a picture of Darwin and quote from The Origin of Species. This quote is explicitly pertinent to this study. And when we think of what he said about congenerics, when he said that the struggle is generally more severe between species of the same genus than between species of distinct genera. So with that in mind, we can then take a sip of our coffee and relax and move to a whole different system, a fascinating system in Southeast Asia. Johannes desmani is a beautiful genus of amazing palms. The entire distribution of the genus itself is depicted here in green. The resolution, it doesn't look that great, but here it is. And then we can break those into different species. There are 4 putative species in the genus, and they're all distributed here in peninsular Malaysia, with 1 species distributed here outside of that region. This is a very collaborative work and came out from a beautiful dissertation published in 2007 by Sue Lee Look. Here she is at Kew on a visit to both John and Bill Baker during her dissertation studies. So when I saw this type of pattern and began talking with my colleagues about Johannes desmania, I started questioning how is this possible? How are all these species sympatrically or parapatrically, or at least co-occurring in this very small region. This region put together is about the size of the American state Louisiana, or about the size of all the Greek islands put together. So I found that very interesting. But maybe what's most interesting is that it's not unique. Unique patterns are more difficult to address. Common patterns might be shedding better light on general patterns. So all of this co-occurrence is certainly not unique, as I just said. Very frequently we see congenerics that are sympatrically or parapatrically distributed, for example in Laquala, in African Marantaceae. And so when we then think of this as more of a general pattern, maybe this lack of uniqueness and overlapping and sympatric distribution of congeners means that we can then extend it to tropical forests in general. And then maybe it means that it's important to explore these types of patterns in a robust manner. So the guys that work on insects had a really wonderful paper recently looking at ecological divergence and how divergence in ecological axes is positively associated with reproductive isolation. So this paper by Funk et al. Showed that across these 8 focal taxa that they looked at, across the tree of life, they found that ecological divergence always increases with reproductive isolation. They found many other results, but I think this is the one that's most pertinent to this part of my talk. So with this in mind, we had various different predictions. We predicted that co-occurrence, or this level of symmetry, was enabled by a lack of gene flow. And that lack of gene flow likely was also seen by segregation along different axes. Is everybody okay? Okay. I'm going to continue. Okay. We can clean up later. We would also have segregation. Let's just clean it up later. Would be associated with ecological divergence. And we also predicted that sympatric species or co-occurring taxa, regardless of, and this is a very important point to make, regardless of the mechanism that formed these species originally. Okay, so I'm talking about current distribution patterns. They likely had differing ecologies. Lastly, we predicted that this co-occurrence likely accumulated over recent geological timescales. So most of the data was generated, by Suli Look. She did an immense amount of work during her PhD. She amassed a morphological data set that is par none to such a small group. She scored 11 characters from over 200 accessions of Johannes Desmania. Here I have the morphological characters listed here. And so the whole goal for the morphological data was that we wanted to know if there was any presence of intermediates, which would indicate gene flow, so that we could look at our hypotheses and predictions. For genetic data, we also developed AFLP haplotypes from over 200 accessions. So 200 accessions from 27 populations across 4 putative species. And the analysis that we conducted with these genetic data was to look at things like genetic diversity, analysis of molecular variants, and population admixture. There was another suite of genetic data that we developed for this project. DNA sequence data was also amassed. We sequenced 204 clones from these 2 nuclear genes across 3 to 4 individuals of each of the 4 putative species. And with these data, we conducted a coalescent species tree and also examined the time since origin. Lastly, we amassed geographic data and performed niche modeling, looking for any type of signal in niche diversification amongst the species using these 4 metrics. We used a PCA, we compared niches, we looked at analysis of variance on their elevational preference, and looked at pairwise geographic distance between each locality. So I had to throw up some palms because we haven't had that many in this talk yet, and these ones definitely deserve a quick minute to ponder before I move on to the results and discussion. Just take a minute to look at this fantastic Johannes Desmania magnifica. It's a huge plant with this white indamentum on the undersides of the leaves, truly making this a magnificent palm. So the morphological data, we treated all morphological data characters together. We did various iterations, but I'm just going to show the major results today. The PCO that we conducted recovered 4 distinct clusters, which are consistent with the 4 putative Johannes Tasmania species that John Dransfield had originally proposed. Also, I'd like to point out that we did not see any significant signature of hybridization or intermediate states using this morphological data. The morphological results were then corroborated with our AFLP data, where using population assignment tests, we also identified 4 distinct genetic clusters using those AFLP data. So once we confirmed that the morphology in the AFLP data showed no sign of intermediate morphology or genetic signature, we then felt comfortable performing a species tree. So this species tree is derived from the 2 nuclear genes that I showed in the methods part of this talk. And as a main tenet of the coalescent, we have to reject the presence of gene flow. So we were able to conduct that study there. So then when we refer to the results in our original predictions, taken together, what we found was that co-occurrence was enabled by a lack of gene flow. Both the morphological and the phylogeographic genetic data showed a lack of gene flow among species. Interestingly, the niche differentiation statistics showed largely similar results for each of the pairwise combinations. As you can see here, in PCO space, these environmental niches are not significantly different. They're highly overlapping. This result was then corroborated as well with between the niche differentiation statistics and the PCO climactic variables, again showing overlapping niche space. So then going back to our prediction, we had thought that sympatric species, again regardless of the mechanism causing their initial divergence, would have differing ecologies. So we did not meet our prediction in that case. And so then when we looked at diversification, we had expected or predicted rapid species accumulation in Johannes Tasmania, as well as the dated phylogeny that I generated for the Livingstonian phylogeny. And what we showed was that Johannes Tasmania had evolved nearly 4 million years ago at its crown node. And therefore, when we make a table, using all of the genera in the palms, we found that it had one of the highest diversification rates, or was one of the most recently involved genera in the family. So again, to go back to our predictions, we did find that co-occurrence had accumulated over recent timescales. And so when we go back to the original goals of this project, was to look at the origin of symmetry, we see that other people have shown similar results. Other people have shown that symmetry originates when there's a lack of gene flow and little time since origin. A colleague of mine from Colorado State University showed similar patterns in Psychotria in the neotropics. When we then get to the maintenance of symmetry, though, the results became a bit more ambiguous because we actually did not find strong ecological divergence estimated from those ecological niche models. Potentially, the niche models don't accurately represent levels of niche divergence in Johannes Tasmania, or maybe the geographic scale at which climactic variables are available don't precisely show what's happening on the ground. So, but what we can say, or when we do begin to speculate, and I do want to warn you all that this part is a bit speculative, that there's no ecological divergence, what's causing the maintenance of symmetry. And then when we review the literature, Len Guan-Sa has published quite a few papers recently on the pollination ecology of Johannes Tasmania. And what you can see here from this paper of Chen and Sa in 2011 is that there are some different aspects of pollination that could be potentially driving ecological divergence. So let's then move on to another case study, another project that I currently have in progress. Discussing a very interesting species in the western Amazon. So sympatric speciation is the formation of species without geographic barriers. It's been shown in very few natural systems. Here we have the Rhagoletus flies. We have cichlid fishes both in Africa and in the Central American volcanic lakes. and everybody's favorite example, Howia. But what is in common in all these examples is that they all are in very highly isolated systems, very unique systems that are either akin or equate to islands. And so what we began to wonder when we started this project was whether the signature of sympatric speciation or differential speciation mechanisms could be detected on a continental scale, such as the Amazon basin. So some of you may be familiar with Johannes Desmania, with Genoma macrostachys from recent papers coming out from our colleague Julissa Roncall's fantastic PhD thesis looking at genoma and specifically at Genoma macrostachys varieties. What she has shown in this immense amount of work that she's put into the system is that there are two morphotypes present across the distribution. We have this bifid, more like leaf in macrostachys, and this more pinnate leaf in the morphotype Ecaulis. So I don't want to embarrass anybody, but Julissa and I have actually been thinking about this for quite some time. It was, these photos actually aren't digital. I had to scan them. But here we are thinking about Genoma macrostachys. In 2003, I was actually a field assistant on Julissa's original field work for her dissertation in Cochacacho, Peru. And certainly Julissa and certainly not myself were not the first ones to be thinking about this topic. I'd like to recognize Henrik Balslev and Finn Borchsenius, who's also been thinking about this topic for quite some time. So altogether, we've embarked on this new journey, this new adventure, trying to find a large phylogeographic pattern in both sympatric and allopatric varieties of this species. So our approach to understanding the phylogeography of Genoma macrostachys is really a trifecta of sorts. We're aiming to understand different processes underlying differentiation using genetics, soil data, and pollination systems. So again, to reiterate, this project is really just starting out, and in many respects it is very large and multi-interdisciplinary and involves many collaborators. So I'd like to just present a kind of a progress report about where we're at, particularly with the pollination biology. So when we look at speciation mechanisms, specifically in the pollination biology, we've been tracking 25 sympatric individuals here in Colombia in Leticia. We have 2 of our 3 field trips completed, and we have compiled data on the pollinator species identification, phenology, although the year is still not complete, and we currently have our floral scent chemistry analyses underway in Brazil. This is in collaboration with Luisa Berta Núñez and our co-advised undergraduate student at Universidad de La Salle in Bogotá. And so what's very interesting, though, is that we currently are only tracking 25 sympatric individuals. So we hope to expand this to allopatric populations in the future, as well as compare this to previous work that has been done, at least in reference, because primarily what's been shown before comes from Ecuador, where when we examine floral scent chemistry, we actually see good differentiation amongst the two. And then a student of Finn Borchsenius has also showed similar results, where we have this mango-like scented one and this lemon-scented one, which is consistent with what Det Knudsen showed in earlier work. And it's also important to complement our results with previous work because we want to understand patterns across the whole Amazon basin. So here's the distribution of Genoma macrostachys from the BN database, and here in blue are those studies, those previous studies, and here we show where our results will be coming from. So we think that we're really going to be able to show some interesting things as pollination biology as a speciation mechanism. So then moving on to the soils, this may be the part that we have the least results on to date. We're currently using Henrik Balslev's results from his Amazonian transects, and when we look at the soil metrics across all of the different types of factors we're looking at, we only really get one positive result, and that's associated with moisture percolation. And so what we still need to do is analyze our 25 collections together with Julissa's soil samples to get a better view about what's happening as per habitat preference in these 2 morphotypes. And again, we want to complement this with previous data that Julissa has published from her dissertation work in Peru, where she explicitly showed that there is differentiation between terra firm and floodplain forests for these two different morphotypes. So we think it would be very interesting then to compare these Peruvian results with that which we find here in Colombia. And then just to finish up, what I'm mostly interested in then is the speciation mechanisms understood from the genetics. So we're currently performing sequence capture on the Illumina platform. I've been working with Jim Liebensmack to work on the sequence capture probes and Perl scripts. And we're targeting currently 174 putative single copy genes. But of those, for example, in Saval, we're retrieving about 100 of those. So we have a lot of data already, and we've done 2 preliminary analyses. The first analysis includes all 75 of the original individuals we're interested in, and we did then a more fine-scale study, which only included 57 individuals that met a particular threshold of quality. Not necessarily interesting to all of you. But within that data set, we have 7 outgroups within Geonoma. We have 17 individuals of variety Macrostachys and 33 variety Acalis. And so this is really the first window into the system where we're looking across the Amazon basin. We have representatives from Colombia, Ecuador, and Peru. And again, we want to marry this with things that have been shown before. Because we've seen some very interesting patterns where habitat drives similarity in genetics. And so it's very important that we bring together all of the previous studies done on this system. So then I just want to wrap up and try to inspire some of you at least to think about future prospects and where you might be going in your own research systems in palms. And so I want to talk about a paper that my friend Alex recently, or actually has still in press, about what they're now calling post-molecular systematics. I don't want anyone to get nervous, but this is happening. Okay, here he's actually saying that the time has passed when a research program in systematics should be based only on a few genes. Okay, this is real and this is happening. And he says population genetics, phylogeography, and species delimitation all benefit from genomic data. This is not just higher level systematics, this is not just Drosophila, this is also phylogeography. It's a big world out there, it's a scary world out there, there's so many techniques, there's so many platforms, what are we going to do? And I think it's really important that we recognize that there's also already been substantial effort put together to make a bait kit for palms. This is the paper that Carolina and Jim Liebensmack currently have in press. This is Carolina here visiting our lab in Barticharta when we had a nice field trip to relax in the midst of writing a bunch of Perl scripts. I'm not going to go too much in depth in this, but I just really want to highlight to all of you that there is a good resource, and it's important that we standardize our results and facilitate beginner success with this probe kit. And just to let you know, there is a GitHub site that Carolina has set up so that everyone can have access to the progress we've made, and we're currently overhauling this. And so some of you might think, well, you know, not me, or next year, but let's just recognize that it's not just our collaborators from Jim Liebensmack's lab or from James Trieger's lab that's been making progress. A lot of people are now thinking and working towards incorporating next generation sequencing in our work. So it's a real shame that Jean-Christophe and Svenning can't be here because him and myself, we very much like oysters. But I just want to remind you that the world is your oyster, okay? Or your palm tree. And that whatever you do, you should feel inspired about the future prospects in the field of phylogeography because there's so much to do, particularly here in the neotropics. So I hope I've been able to give a good overview of the current state of the field. I've tried to focus on different taxonomic levels, highlighting a few case studies in palms. And with that, I want to thank the members of my lab, particularly my supervisor, Alex, and my PhD co-supervised student, Alex, as well. And I also need to recognize my funding sources and all the co-authors here on this work I presented today, as well as you, the audience, for being so receptive and understanding for my frantic state of baby Dim, and also the World Palm Conference organizers. It's been such an exciting event. Thank you.